All right, our next talk is going to be from Leonardo Ferreira from the Medical University of South Carolina, and his talk is entitled Creating Localized Immune Tolerance in Type 1 Diabetes Using Chimeric Antigen Receptor Regulatory T Cells.
Good evening. I hope everyone is having a fun and productive meeting thus far. A big thank you to the organizers for the chance to speak to you today. So we like to think of our immune system as an army that goes out there and protects us from viruses and pathogens. But sometimes that army goes rogue and actually goes at war against ourselves. That's autoimmune disease. The nearest and dearest autoimmune disease to our hearts is type 1 diabetes, where beta cells get progressively destroyed by mostly a T-cell-driven response. So we have a scenario there where your pancreas is pretty much on fire, thanks to these autoreactive T cells that recognize beta cell antigens. But not all T cells are evil. There are these regulatory immunosuppressive T cells. They're characterized by a high expression of CD25, which is a high affinity receptor of IL-2, and they were first discovered in mice, where if you thymectomize mice early on, mice will get oophoryditis, thyroiditis, and type 1 diabetes. So we knew there's a regulatory T cell in there that needs to develop very early in life. If you get rid of it, you get autoimmune disease. Another point is that these cells express FOXP3, transcription factor. If you have humans that have FOXP3 mutations, they have all sorts of autoimmune disorders, including, in 80% of cases, type 1 diabetes. So now we have this emerging picture where you have this subset of T cells that develop in the thymus, and they express FOXP3 and high CD25, and they go out there and they ensure immune homeostasis such that nothing goes overboard in our immune system. There are two main tenets for our regulatory T cell, or Treg, works. One is bystander suppression. So when a Treg gets activated in a local environment, everything around it also gets suppressed. So T cells against other antigens will also be inhibited in this microenvironment. Number two is infectious tolerance. So we like infectious laughter. After the Treg is gone, it convinces other naive T cells to let themselves become Tregs. And so the hope there, this is yet to be proven in humans as rigorously as in rodents, is that if you infuse Tregs and they have a certain lifespan, their effect could live on after those injected Tregs have disappeared. Multiple molecular mechanisms through which Tregs work. They sometimes speak directly to the manager, the antigen-presenting cell, and emasculate them by stealing CD80 and CD86, so they can no longer activate T cells. They can also directly kill APCs via perforin granzyme B. And sometimes they just rearrange things. For example, they take ATP extracellular, which is a danger signal, and convert into adenosine, which basically puts T cells to sleep. So it's kind of a polypharmaceutical, hard to replicate in one or more molecules. So we think Tregs are a prime candidate to go in cases where we have excessive immunity, to tilt the scales and go into regulation. So does this really work? In rodents, yes, it's been shown now 20 years ago. They have a non-obese diabetic mouse, and you infuse as low as 500,000 antigen-specific, islet antigen-specific Tregs, they can prevent the occurrence of type 1 diabetes. If you take mice that already have type 1 diabetes, you now infuse 10 million antigen-specific Tregs, and now they can actually subvert, they can reverse this disease. So you know that in principle it's there. And importantly, they have to be antigen-specific. You take just polyclonal, bag of specificities Tregs that don't do anything. You see the glucose just going up. It's just the same as infusing PBS. So you have to have the BDC 2.5 Treg, which recognize an islet antigen. So how do you produce this in humans? Well, not polyclonal Tregs. It just came out 20 years after that study that in humans, if you also infuse polyclonal Tregs, just like the mouse, also no protection. It was a 110-patient study with a high dose, low dose of Tregs. So we need an antigen specificity. But antigen-specific Tregs are rare. It's only a few per million cells in peripheral blood. Sometimes we don't even know what they should be seeing. So that's where we took a page from the cancer world and used what's called a chimeric antigen receptor. This basically puts together T-cell receptor signaling, or signal 1, and co-stimulation, which is usually C28, signal 2. So in a single polypeptide, you have the whole machinery to activate T cells, and an extracellular domain is antibody-based. So now any surface antigen I can think of, I can redirect my Treg to go there and get activated upon exposure to the antigen. And so the point of application for this will be the current functional treatment for type 1 diabetes, as we heard, which is islet transplantation. From a deceased donor, you take islets, digest them in a Riccardi chamber, put them in a portal vein in the liver, and there they are. Or you can also take stem cell-derived islets, or islet-like cells, and also put them in a portal vein. So this is the idea then. So you're going to address the loss of beta cell mass by putting more beta cells in. But if you do it alone, it's like building a castle, sand castle by the ocean. It's going to be washed away. So you've got to protect the castle putting it in. So first then we wanted, what should these CAR Tregs recognize? So a place to look for unique antigens is HLA. There's thousands of flavors of HLA. And so we wondered, one example to use this is, let's take HLA-A2 positive donor islets that are going into an HLA-A2 negative recipient. So now HLA-A2 becomes a unique marker for the transplanted islet. So we started there, and we generated an HLA-A2 CAR and put in Tregs such that another tiny firefighters that go straight into the inflamed pancreas that would stop that autoimmune attack, which then will allow for these transplant islets to work. So the way we did this, we tried to be as precise as possible. You now know CAR T cells have this black label problem that they could cause cancer. So now we actually knocked in the TCR locus, which is what T cells like to use to see the world, and we knocked in this HLA-A2 CAR. And what we see is that the Tregs, the knock-in works, right? They are MYC-TAC positive. It's a marker on the surface. They lose CD3 expression, so they replace. They have no HLA-A2, of course, otherwise they would talk to each other too much. They steal Tregs, so FOXP3 and Helios, double expression, is a naive, well-behaved human Treg. And of course, they get activated by exposure to the antigen, which is in the case HLA-A2. And we look at this by looking at CD71 upregulation on the surface of the Treg. So we do this, so now in vivo, do they go to where I want them to go? And so to do this, we took NSG mice, the lucky winners of the lottery. We gave them diabetes with streptozotocin, so they lose their endogenous beta cells. Then we take cadaveric islets, put the mouse on its side, expose the kidney capsule, and put the islets, the human islets, HLA-2 positive islets, under the kidney capsule, suture it all back together, wait 3 weeks or longer, and then we do an immune challenge. In this case, we're going to have luciferase-labeled HLA-A2 CAR Tregs, or polyclonal controls with just luciferase. And so what we see very clearly is that at the beginning, when you infuse these cells in, tail vein intravenously, polyclonal Tregs and CAR Tregs, they all start their journey at the spleen after you go into the mouse, but then over time you see the polyclonal Tregs, they just lose their way. Whereas the HLA-A2 CAR Tregs migrate, they traffic to the HLA-2 positive graft, and they stay there. Now we wonder, they're there, are they doing harm or anything? And the answer was no. Remember, these mice depend on these human islets to remain normoglycemic. And so what we see is that if you infuse these mice with HLA-A2 CAR Tregs, the blood glucose does not change. So these are safe, so it's feasible. So you reproduce what we see in humans, polyclonal Tregs, the same in the mouse. Now, of course, 2 questions here. One is, we don't know if they actually protect from immune attack. That's one question. And number 2 is that not everybody is HLA-2 positive or receiving an HLA-2 positive transplant. So maybe we want to enlarge our horizon and come up with our own targets that CAR Tregs could see. And so this was work done with Rob Rubino in my laboratory, research specialist. He's now actually at UPenn as a grad student, first year. So UPenn people, you can still grab him. He's an extremely hard-working student. And in collaboration with Holger Russ and Jesse Bauer, a postdoc in Holger Russ's lab at the University of Florida. And so what we did was, we started from stem cells, made them all the way to beta-like cells. And so this, as we know, is an alternative strategy to cadaveric islets for transplant. But again, they function, but they still have a challenge of they need to be protected. So maybe the CAR Tregs can protect them as this polypharmaceutical cell that can protect them from all sorts of unwanted immune responses. But then, of course, what do they target them to find a unique target? And so we make our own. So we sat down and thought, how are we going to go about this? We need a target that is stably expressed on the cell surface. It's not going to be trafficking, cleaving, all this. You should be biologically inert. You don't want to hurt the beta cell you want to protect. You should be immunologically inert. If you put some molecule from a different species, it might cause rejection. And it should be unique, not expressed by other cell types. So all these categories, our first attempt was then using a truncated EGFR. So if EGFR itself is not immunogenic, neither should be a fraction of it. Because truncated, there's no intracellular domain, and it's also missing an outside domain there. So this is our proof of concept unique ligands to be on the surface of beta cells. And then concomitantly, we took Tregs, made them into CAR Tregs specific for EGFR. So we're basically making the lock and the key of this arranged marriage of these cells. So for the stem cells, what I show here is that we knocked in the AAVS1 locus, the EGFR2, the CAC promoter, and a luciferase gene for later on track them in vivo. And so we show that this doesn't affect the stem cells. They have OCT4, they have NANOG, they express EGFR2 on the surface by flow, and they also luciferase activity when they get the substrate. Now, in terms of their function, we see it does not affect their C-peptide levels. So in the graph there, they have the same exact percentage of C-peptide production, and they maintain EGFR. So the beta cells maintain EGFRT expression on their surface. They have the target. Their function, again, is the same. If you simulate them with glucose, they have your typical GSIS pattern with glucose. Now, on the immune side, we take Tregs, transduce them with lentivirus with the EGFR CAR. You see very high transduction efficiency. They remain Tregs, FOXP3 Helios high. And you see that compared with the T effector cells control that don't have it. Now, first things first is, do they talk to each other? So to do that, you look again at C71 expression. So you see, when you co-incubate EGFR CAR Tregs with EGFRT expressing SPCs, you see upregulation of C69, C71. Also, they make cytokines. They make IL-10 and IL-13. Also, curiously, they don't seem to affect the beta cells' properties in chemokine production. So these beta cells make MCP1 just fine, with or without the presence of CAR Tregs. We also went to great lengths to make sure that these CAR Tregs are well behaved. So one thing is been shown that Tregs can destabilize if you activate them too many times. So we did 1, 2, 3 times. Every time the cells are 80% and above FOXP3 Helios double positive. And if you co-incubate them with SPCs, they do not kill them. Zero cytotoxicity with EGFR CAR Tregs. Also, no inflammatory cytokine production. Gamma, TNF-alpha, IL-17, you see zero almost production of these inflammatory cytokines. Okay, so time to show some function. In vitro, two very popular assays. One is to show that CAR Tregs inhibit T cell proliferation, and you see here in CAR CD4s and CAR CD8 T cells, suppression proliferation. Also, as I mentioned in vivo, Tregs love speaking to the manager, so we want to make sure these CAR Tregs are inhibiting dendritic cells. So we took blood monocytes and made them into dendritic cells, and then we show that the CAR Tregs are decreasing CD80 and CD86 on the surface of dendritic cells. All in vitro, so we're ready to go. So now, in vivo experiment is you take the mouse, kidney capsule transplant, and then you do the immune challenge after engraftment. You do either CAR T cells alone, which will reject the graft, or CAR Tregs and CAR T cells, and see if now these CAR Tregs protect from rejection. And so what we see here is that with luminescence is that indeed the CAR Tregs protected as based on graft of the mass of the graft. So you see the significant difference there. Now, I'm an immunologist. I crave and need flow cytometry. So my first question was, are the CAR Tregs in the graft? So took the kidneys out, digested them, did flow, and you see very nicely, you find human CD4 positive, M-cherry positive, FOXP3 positive cells in the grafts of the mice treated with CAR Tregs, but not the controls. But then, of course, seeing is believing. How about what the cells look like? So then, again, Dr. Russ's collaboration, this is probably the most important picture in the talk, is that you see, if you have the effector cells alone, very few and far in between human insulin-producing cells, a lot of T cells, CD3 positive, not really many Tregs. But if you have the CAR Treg-treated mice, you see beautiful clusters of insulin-producing cells. You see some T cells, yes, but most of them are FOXP3-producing Tregs. And so you see that here. And then you also see that they maintain EGFR production, so it's not the target gets lost along the way. And here's also an overlap where the same cells that express insulin also express EGFRT. And here's a quantification where you see that basically 50% of the T cells in these protective grafts are now Tregs. So we made the lock and the key of this localized immune protection of these stem cell-derived beta cells. So we got ourselves a mix of an off-the-shelf and a personalized potential treatment for type 1 diabetes. So off-the-shelf, we engineered stem cell-derived beta cells. Again, we did not engineer any HLA, 347, anything. Only this ligand that is inert on the surface. And we take the patient's own Tregs, engineered with a CAR, to see that ligand. So I hope I showed you that these CAR Tregs, they not only traffic to the expressing graft, they actually protect the graft from immune rejection by these T cells. So very quickly then, what's next? One thing that was brought up was we need better models. How about the B cells? How about the NK cells? How about other T cell subsets? So the state of the art is this NSG mouse with 4 human genes knocked in, stem cell factor, GM-CSF, IL-3 and IL-15 that better supports human myeloid cells, human B cells, and human NK cells. So we can use those instead of the current NSG mice. Then, of course, people don't have CAR T cells in their blood. So we would use hepatogenic T cell receptor, engineered T cells as rejectors. And then, of course, third is we would use STZ as a way to try to mimic the inflammation that can happen with hyperglycemia in these mice. That's one aspect. The other one is that EGFRT might not be the best target to go into humans. So can we make our own target that is really unique? And so part of my lab works on cancer, and as usual for me, didn't work out that project. But I took that target as like, I'm going to take this target and make it into a unique cell surface obligatory ligand. And so what we have right now is it really seems that I was able to force this molecule to go on the surface of target cells. We also make an scAV for a CAR that binds to that CAR, and so that's where we are right now. We have this new ligand. But then, of course, this CAR can be just anything. We also found in the lab that CAR Tregs can be a bit picky. So usually a T cell receptor, the residence time in antigen is 3 seconds. It's a kiss and run. The CARs that we've used, some of them are 3 minutes, and one of the lab actually is 3 hours. So more like a French kiss. And so what we see is that these CAR Tregs, look at that, interferon gamma, TNF alpha, CD40 ligand being shed. So you need to be very, very careful for CAR design, specifically tailored for Tregs. And so there's actually collaborative lab at Duke that is using artificial intelligence, protein language models, they call it, to be very, very judicious where in the protein it binds and with what affinity it binds with. So we're now able to generate different affinities at different sites for our new CARs. And a quick idea is that, of course, we don't live under the rock, so no, there's an orthogonal approach to protect the cells, make them more immune evasion. I myself used to be part of this world. And so I think there's great potential there to synergize. Maybe some edits that help protect the stem cells can be synergized with having Tregs there at will that will give localized protection. And so with that, I think this can be maybe a total off the shelf. Recently just came out from a Japanese group that they can actually make Tregs from stem cells. So I envision a future where we can take stem cells, make them into Tregs and then CAR Tregs, and into stem cell-derived islets. So everything will come from the same tube. And so with that, I'll leave with some outstanding questions. There'll be unique challenges for CAR Tregs in T1D, hyperglycemia, IL-2 deprivation, insulin. Tregs actually hate insulin. They have insulin receptor signaling. We can talk more about that. The other one is what's the best source for the Tregs? The same person, different person, stem cells. And so there will be a few questions here that we can address as a field. And so with that, very important slide. I need to acknowledge people in my laboratory. We've had a lab for 2.5 years now. Also some work I showed still at UCSF with Dr. Tong and Dr. Bluestone, collaboration with Dr. Russ and his postdoc, Dr. Barra. Also, I collaborated with USC, Clemson, and Duke, so I really embraced the South in pushing this field forward. And, of course, the funding. One of them being, I was one of the 5 recipients of the HERN Emerging Leader in Type 1 Diabetes. That was actually my first grant ever. I got my green card in May 2020. And I applied that same month. And so I'm extremely thankful for how much HERN has helped me to launch my own career as a principal investigator. Thank you.
Leonardo, beautiful study.
Thank you.
Just want to share with you some of the studies that you have done parallel to what you've done in the mouse. With Carl June and Jim Riley, we created Treg specific for HLA-A2, except that we used non-human primates. And I'd like to share the challenges that you have. The challenges were when we infused the Tregs, we don't know where they are. So we really surveyed the entire immune, anatomic immune. Interestingly enough, all the Tregs were collected into the bone marrow. Then we came upset, well, if they need to be in the proximity of the allogeneic islets, these are not a stem cell. We actually learned from the Italian group 10, 15 years ago, where I injected the islets instead of into the liver, into the bone marrow cavity, so they would be in the proximity of the HLA-A2, and it didn't work. These are really basically correcting hyperglycemia. We also, of course, thought about the IL-2. So we contacted Chris in lab, and we got some orthogonal IL-2. Okay. Because, as you know, the Tregs depend on IL-2. I can share with you that also turned out to be negative. So I want to make 2 comments. One is I am a really a student of Tom Waldman's infectious tolerance, but I think that concept, at least in my knowledge of transplantation, not autoimmunity part, has not been extended beyond the rodents. And finally, I think everyone knows here, the mice experiments lie to you, but the non-human primates don't tell you the truth. So I don't know where we stand.
These are excellent points. I know there are efforts on both addressing the IL-2 deprivation, also the chemokine receptor. So I think this is version 1.0 of engineered Tregs, where we're just changing what they see and then hoping that the Treg does the rest. But it might need some help. It reminds me of the solid tumor world, where now they're expressing, in the CAR T cells, they're expressing CXCR1, CXCR2, because monocytes expresses receptors and infiltrate solid tumors. So likewise, we might want to, in addition to the CAR that provides antigen specificity, also express some chemokine receptors in Tregs that ensure they don't go to the bone marrow, as you mentioned, but instead to the graft, wherever it might be. So I think that's an excellent point. And the other thing is, I agree, there's no rigorous, it's hard to prove infectious tolerance in humans, right? Because if in a mouse we can serially transplant and wait a year or two, in humans it will be 10 or 20 years to prove that indeed that infectious tolerance happened. But it can be done, but it has to be a 20-year-long study to find out. So at this point we don't know. But I think that in the end, I'll say just this one more thing, is that it has to be a combination. So I think we should get any help we can get. We might have to further engineer the Tregs, make them more resilient, more likely to migrate to the place. We can engineer, if the stem cell-derived beta cells really work well, that's a whole new platform to engineer, because stem cells are relatively facile to engineer the genes, so you can add some genes that can further help. And I'll say on that regard that, again, it's been a big focus on, and I've used myself, knock-out MHC class 1, knock-out MHC class 2, knock-in HLA-G, C47, that's it. But I think we could do further edits that can get into other pathways. So actually from the pig organs, we can learn quite a bit, because in those people, they added the complement receptors, they added oxygenases and other enzymes. And so I think we can put in more edits on the stem cell-derived beta cells to prolong their survival, such that we even stand a chance against a very strong autoimmune response and a very strong or even stronger alloimmune response.
Steve from Cornell. Nice talk, beautiful work. I have one question. Imagine you're going to use this in human, and if the patients who received, let's say, SC islets have allogenic rejection to the SC islets, and you're going to use the CAR Treg to suppress the localized suppress the immuno attack. Do you need multiple doses, or do they proliferate, or do they live, like, do the Treg live there in the body for a very long time? And how would you imagine this?
Yeah, it's a good question. I'll say in vitro they proliferate upon antigen exposure. In vivo might be a different story, but we know we went out to a month, and when we slice through those grafts, we see FOXP3 positive cells there still. Now, it could require multiple infusions. I think the good news is that because CAR Tregs traffic to the organ, it is feasible to do multiple infusions. It's not like you have open chest surgery every time you need to put in more Tregs. And so I do see a scenario where you might need to get Treg infusions every month, every 6 months, every year. We don't know yet. That's one aspect of this. And then the other one, again, as I mentioned, there might be other edits in the stem cells that can synergize with it. But I think it's quite a strong response we're against. And so maybe Tregs are not enough. And in the field, people combine with rapamycin, combine with low-dose irradiation. And so in my ideal world, I would like us to engineer super Tregs, so that the CAR not only redirects the Treg, but also makes them even more resilient than normal Tregs. I think the suppressor cell works in that direction, where a lot of TGF-beta, a lot of C25, and see, can we kind of break down what is the minimum setting that a Treg needs to be really very active. And so I think it's exciting work for the field to figure out these rules of the game, if you will. They can then make a better and better version of these living therapeutics to protect the islets. Yeah, thank you.
Thank you so much.
Thank you.